disorders. Um, So we hope that these efforts will result in the identification of novel gene targets as well as novel therapeutics for hearing instability disorders. And currently, really, the only thing we have for the treatment of these disorders that has kind of any real strong proof are steroids. There's some other adjunctive treatments that, from antivirals to hyperbaric oxygen therapy, that have very small amounts of evidence and aren't widely available necessarily. So to take a step back, the Stria Vascularis, what does it do? So it generates and maintains something called the endocochlear potential. It's essentially a battery for the inner ear. Richard, can you see my cursor? Can you guys see my cursor?
Yes. Yes.
Okay. So the stria is a three-layered structure, and it's predominantly composed of these three layers. The first layer composed of marginal cells in pink here, the second layer, the intermediate cells, and the third layer, the basal cells. Mixed within these are a bunch of other different cell types, and all these cell types work together to generate the endocochlear potential, as well as to kind of transport and concentrate potassium ions as well as other ions in the scala media and the endolymph, which is contained in the scala media. And what we know is that this endocochlear potential is really necessary for normal hair cell function and thus hearing. So we know that diseases from like human temporal bone studies, that diseases of hearing instability and hearing fluctuation have been associated with endolymphatic hydrops, and that's this ballooning out of that scala media, primarily into kind of the scala vestibuli. And we also see thinning of the stria in some of these pathologies over time. But what we're seeing in human temporal bones is kind of the end result. And we don't really have an idea of what's going on kind of in the midst of that disease during the lifetime of these patients. And really, no effective treatments really exist for hearing loss in these disorders. And it's thought that the hydrops that we see reflect some dysfunctional endolymph homeostasis, homeostatic process in the cochlea. So, you know, some supportive evidence for this is that, for instance, in Meniere's disease, you know, these patients, when we've looked at them post-mortem, they exhibit largely intact organ accordi. So the hair cells and the supporting cells look fairly intact. Now, some of them may have some loss of outer hair cells, but and late in the disease, they exhibit some loss of hair cells. But earlier in the disease, and sometimes even throughout their disease, these patients have fairly normal appearing cochlea or organ accordi. So we think that the disease does not necessarily start with the hair cells in Meniere's disease. So, and others have looked at other human temporal bones, and specifically at the Stria Vascularis, and shown that there are changes in the Stria Vascularis as they relate to Meniere's disease. And these are kind of distinct from what we think of as age-related declines or structural changes in the Stria Vascularis. So, so this is kind of just bringing you back to kind of what we've been doing. And what I'm going to talk to you about is some of our initial efforts in really characterizing the Stria Vascularis using transcriptome studies. So we have been using single cell and single nucleus transcriptional profiling to take a look at the individual transcriptomes of each of these cells. And in the past, gene expression profiling has been utilized to make connections between gene expression and function. And the starting point for this, and it's often served as a starting point for poorly understood diseases in complex tissues. And the starting material has often been whole tissues or populations of cells. And here you see kind of two areas of interest from my lab, the stria and the supporting cells in the organ accordi. So what's the difference between looking at things from a whole tissue standpoint when you're looking at transcriptomes or RNA that's expressed by those cells and tissues, or looking at single cells? Well, you can think of the tissue or populations of cells as kind of like looking at things as if you were looking at the results of a milkshake or a fruit smoothie. You get kind of an average. You know, everything's put into a blender and you kind of see an average of what's there. When we look, kind of recent technological advancements have allowed us to kind of take a look at single cells now. And what it's allowing us to do is look at things more like as if we were to look at things like they were part of a fruit bowl. And we can look at each piece of fruit and taste each element, and we get kind of a more accurate sense of what's going on in the tissues that we're studying. So just to give you an idea of what the difference is, again, to emphasize the point between what the difference is between looking at a population or tissue and looking at individual cells. So in a population, we may get this average of 3 different genes that we're looking at, okay? But if we look at individual cells, each individual cell may have a unique combination of those genes. So the cell type specific profiles are not necessarily apparent when we look at these populations or averages. And even more important is that rare cell types, which may contribute to disease that may not be evident from these expression profiles. So just to give you an idea of the pipeline of how we do this, and we published a review this year. Richard, you're gonna have to tell me if anyone's asking a question along the way. I'm happy to answer anything along the way. This doesn't have to be just me talking.
No problem.
Okay. So, so cells or nuclei are separated from, from these tissues, and then they're captured either through droplets or microfluidics-based technologies where the cells are captured in droplets, or they're deposited in wells. In our case, we did the droplet route here. And what happens in each of these droplets is that the cells are lysed and we extract the RNA and we convert that to cDNA. And I know this is reaching back to some science that some of you may or may not remember, but this allows us to study the RNA that's expressed by these cell types. And we take this RNA, the cDNA, and create these libraries so that we can know all the RNA that's expressed in that given cell, or at least a good percentage of that RNA. Okay. And there's some bioinformatic processing that occurs afterwards that allows us to then look at this data in a format that we can understand and interpret. And basically down here, what you're seeing is each of those colored groups of cell, each dot is a cell, and each of those colored groups is a different group of cells that share certain groups of genes, share in common certain groups of genes. So we're able to distinguish cell types by the genes that they express. And this technology is enabling us to not just understand more about native tissues, but also to understand what might be happening in disease. At least that's what we're beginning to think about, to utilize these technologies for. So, anyone who has any questions along the way, or if there's something you don't understand, please ask. I'm happy to stop. I made plenty of time so that questions can be asked here. So what have we learned from using these techniques about the Stria Vascularis? And that's really what my lab has spent the last couple of years really delving into. So, so we've taken, used the adult mouse in terms of the data that I'm going to show you today. We've used the adult mouse and looked at the Stria Vascularis and looked to identify the transcriptional profiles of these individual cell types. So we have characterized the transcriptional profiles of marginal, intermediate, and basal cells, notably. So you see them here in pink, green, so green for the intermediate cells and blue for the basal cells. So pink's the marginal cells. So we've been able to do this both with single cell and single nucleus transcriptional profiling. And we've identified some other cell types along the way, some cell types like the spindle or root cells in the stria, which are these little yellow cells here, as well as kind of the fibrocytes in the root cells here, which we also think that play a role in potentially a role in hearing instability. Okay, so as I stated before, we've used these single cell transcriptional profiles to establish kind of known expression profiles for these cell types. And previously, we didn't have all this information. And while this may seem like, I've kind of made it very simple, but this may seem like this is something that we should have had all along. We just haven't known this. I think having these kinds of databases is now allowing us to potentially reference, genes that people have studied in the past and trying to understand what cell types might be involved. So we're using these databases that we've generated to understand diseases now like sudden hearing loss and Meniere's disease and other genetic forms of hearing loss. So this is just an example. So these are all considered known markers for these cell types here. So in this column here, these are known markers for or known genes that are expressed by marginal cells, and you can see that the more blue a dot is, here you see they're kind of purple. The more blue it is, the more likely, or the higher that the higher level of expression that gene is in each single cell. And I should take a step back. So each of these plots are called UMAP or TSNE plots, and each of the dots represent a cell, and we've grouped cells together by colors, and this is done based on their shared expression profile. So we don't choose them ahead of time. The genes that they express allow us to determine that after the fact. So it's not, but what this tells us is that that kind of analysis is valid because these groups of cells express genes that we know are expressed by those cell types. So marginal express.
Mike,
go ahead.
Let me ask you a question. Okay, we know Meniere's disease or endolymphatic eye drops has, an increase in the in the endolymph of the scalum media. So when that happens, it compresses the stria vascularis, by a mere volume effect. That changes the action or the battery component, action potential of all the cells. So your sodium potassium pump's not working, correct?
I don't think we know that exactly. I don't think we know where. There's been some suggestion that the fluid pressure, that the pressure exuded by this kind of expansion of the endolymph, that it acts on either the hair cells. We don't know what it does to the stria. That people have not measured that in those settings to understand that that occurs. That is conjecture, really, and it's based on, I think, human temporal bone studies, which suggest that structures are compressed or that structures atrophy, but I don't think we truly know that.
Okay, so you're getting you're looking at the marginal, intermediate, and basal cells, and you're looking at the different genes that are being expressed. Tell us more as to what are you thinking when you're looking at these genes?
So we think that so what we're looking for, really, in the end, we want to understand more about the mechanisms, and there are groups of genes that work together to accomplish function here. And so we see that within cell types, that they have these groups of genes are called gene regulatory networks. And so that these regulatory networks underlie function in the Stria Vascularis. And we think that under certain situations or pathologic conditions, like Meniere's disease, the function of these regulatory networks is altered. And the alteration of these regulatory networks can result in the mechanisms that regulate fluid and ion composition in the cochlea could be affected, as well as the ability to generate this endocochlear potential. So we think these mechanisms are affected. Now, what we're doing here is really setting a basis for understanding what those changes mean, because we are looking at an animal that is unperturbed, that does not have hearing fluctuation. It's considered a wild type or a kind of baseline characterization. So in order to understand how things change, we have to understand where they are at the beginning. And this is what we've tried to set out with these studies, because before we really did not know that. We didn't know what was there. And we knew some of what was there, but didn't have a good sense for the lay of the land. So these studies are an attempt to understand what the baseline is, and going from here, our group is using this baseline knowledge to look at situations where hearing is fluctuating in animal models and in humans, and trying to understand how these regulatory networks change, and how that relates to hearing instability and hearing loss. So this is just showing, looking at we're able to localize the RNA that's expressed by these cells. We're able to localize them to specific cell types. So this is just showing that, so these are two genes that we know are expressed in marginal cells, and each of the dots here to the right on these images, this is an adult mouse, Stria Vascularis, and each little dot, whether it's red or green, represents a single or a couple molecules of RNA. And this is the marginal cell layer. So, and any of the white are the nuclei of the cell. So the marginal cell nuclei lie on the surface. So we can localize both KCNQ1 and KCNE1 RNA to these cells. And we've done this with several novel or new markers, and been able to kind of provide confidence in the data that we've generated. So basically, these validate the fact that the data, the resources that we've created can be utilized to kind of understand what genes are expressed. And we've also looked at some of these genes in human temporal bones, and this, this data is consistent in humans to a large extent. So it's definitely using this mouse, these animal model systems, we can reference this to human genes and get a sense for where things might be expressed in the human cochlea. Because unlike animals, we don't have necessarily ready access to human inner ears. So this gives us kind of, allows us to get some insight into what's going on in humans. Okay, and I'm going to skip this. These are just some new candidate genes that were confirmed in the adult Stria Vascularis. These had not been previously known in this study. So these were some of the novel findings of this study. This is just one example of that. We're able to kind of localize a given gene that we've identified to intermediate cells. Here, what you're seeing, the novel gene is, the novel gene RNA is in red, little red dots, and they're in this intermediate cell layer of the Stria Vascularis. But I think what's more exciting to us, and what we're working on now, and is really looking at these underlying gene regulatory networks and how they relate to hearing function. We think that these regulatory networks form the foundation for relating dysfunction in the stria to hearing instability and hearing fluctuation. And you can think of these regulatory networks as there are master factors or transcription factors that activate several genes, and we're looking to identify these factors and the networks of genes that are involved, because we think that these relate to kind of larger functions or major functions in the stria. And we've done that here. What you're seeing here is kind of a plot showing these regulatory networks that we've identified in given cell types. So basal cells here, marginal cells here, and intermediate cells here. So, and we've looked at, I'm going to give you an example of a given regulatory network. So we've looked at an estrogen receptor related, estrogen related receptor beta, which there's some publications and some data in the literature that suggests that estrogen may be protective of hearing. And we've utilized druggable targeting databases. So looking at databases that look at drugs that are capable of modulating gene activity or altering gene activity. And so we've utilized our gene regulatory networks, and this is an example of that, of one of those gene regulatory networks, and found that that several of the genes in the pathway have drugs that are capable of targeting those genes. So, you know, I know that many of you are living with and dealing with COVID. One of the things that they're doing now with COVID is they're employing very similar drug repurposing approaches. So they're looking at genes and structures of the virus and seeing what drugs can be used to target those structures and genes. And they're finding drugs that can be used that are FDA approved, but that can be repurposed for treatment of COVID. So we're interested in kind of a similar approach to some degree, and we're looking for druggable targets for hearing fluctuation and hearing instability. And just an example or an application of our databases, we can take a look at a list of known deafness genes and show basically where are these known deafness genes expressed in the Stria Vascularis. And we can relate cellular dysfunction or dysfunction within given cell types in the inner ear to certain kinds of deafness. So these are all examples of diseases where a mutation results in deafness. So we can definitely see that the stria is important for certain kinds of deafness, and I think that's what this highlights here.
That's pretty amazing when you start really thinking about it. I mean, we're getting to the cellular level, and we're starting to, at least begin to understand the frontier of hearing loss, at least, congenital hearing loss.
And we hadn't known that before. And, I think, one of the things that we do know, so what I'm not showing here is, so connexin 26. One of the things we know is that connexin 26 is, based on our data and others, is expressed by both intermediate cells and basal cells, as well as supporting cells. Okay, and we know from some work done in animal models that suggests that the problem with our gap junctions may lie in the fact that they're missing in the stria, and may not be a result of them missing necessarily from the supporting cells. So, but nonetheless, these atlases or these data sets that we've, as a field, have begun to kind of construct and provide to the wider scientific community are allowing us to figure out where to target our therapies potentially. And connexin 26 mutations are probably the most common genetic mutation that results in progressive sensorineural hearing loss in kind of humans. So in terms of why did we use two approaches, and I think I definitely glossed over that in the beginning, and one of the reasons I want to talk about this is because the approaches that we're utilizing and using single nucleus approaches, they can be applied to human tissue a lot more readily in some ways. And I think that it's going to be in this route that we might be able to get at other diseases that we have a lot of problems with finding treatments for. So what are the advantages and disadvantages here? So from our standpoint, with both techniques, we get a lot of cells, but in tissues where there's a lot of cell size and shape differences between the different cell types, it's a little easier using the single nucleus approaches because the nuclei are fairly uniform. Now, I think one of the things that is more relevant to human tissue is that the single nucleus preparation is fairly easy on tissue, and we have ways to preserve those nuclei or preserve the tissue so that they're still amenable to nuclei approaches. So meaning tissue can be shipped in certain kinds of preservation material, and then the tissue can be analyzed using these techniques. So certain kinds of tissue are harder to dissociate or break into single cells. And the nuclei approach allows for kind of an easier ability to do that. For instance, in the stria, the cells are interdigitated with each other. So the marginal and intermediate cells kind of interlace with each other. And that makes them difficult to separate. But the nucleus preparation allows us to overcome some of these. And you can think of some other areas where this is applicable. Human tumors are a good example of this, particularly schwannomas. Also, tumors in the brain are also kind of at issue, and certain kinds of salivary tissues are also good examples of tissues that are hard to dissociate. And finally, one of the things that we're very interested in are these gene regulatory networks, and this is a relative advantage because we've found ways recently to overcome some of the weaknesses of single nucleus in terms of identifying gene regulatory networks. But I think one thing I will point out to you is that our approaches with single nucleus allow us to really better separate cell types and kind of get a better lay of the land when it comes to both normal tissue and kind of pathologic tissue, including tumors. So in summary, I've shown you that we've characterized the adult mouse Stria Vascularis cell type specific transcriptional profiles, and we've identified both known and previously uncharacterized cell type specific gene expression. We've identified some homeostatic gene regulatory networks, or genes that underlie function in the adult Stria Vascularis, as well as identified some druggable gene targets. We demonstrate a possible role for adult strial cell types in human deafness, and demonstrate the complementary roles for single cell and single nucleus approaches to RNA-seq in the adult stria. With that, I'd like to acknowledge everyone in my lab, as well as my collaborators who've contributed to the work here, and highlighted are the people who have accomplished or contributed to, made major contributions to the work. And thankful for my research funding too. So with that, open to any questions.
Mike, let me ask you a question. How many different institutions are doing this research in conjunction with you at the NIH?
So, so I have several collaborators. The work on the stria is being done primarily by my group. We have collaborators that we have been doing work on looking at strial macrophages or cochlear macrophages. And we have some collaborators that we're working together to look at human tumors like vestibular schwannomas. So we're definitely interested in kind of applying these techniques more widely. We have a publication that is under review where we're talking about how some techniques that can be used to preserve samples and successfully obtain these kinds of profiles. But that's in mouse, and then we've done some of this work in humans, but we haven't published that yet. So.
Great. Anybody have any questions? Feel free to unmute or raise your hand. Lovera, you have any questions from Paraguay? Marta, do you have any questions?
Yes, please.
Okay, Marta from Barcelona, Spain. You know.
Yeah.
Thank you for your presentation. I would like to know which kind of approach do you have with this research that you are doing into the clinical practice? And will you explain the fluctuating hearing loss due to the Stria disease? And if you only can use corticosteroids, or you have thought, or do you know if anybody is using like immunosuppressors, like methotrexate?
So we've looked at, those are all really good questions. So let me start from the top. In terms of how we're applying this to the clinic, one of the first things we've done is to try to apply this to human tumors, because we can get a lot of tissue. Getting a lot of tissue from the human inner ear is a little harder. So we've done these with some vestibular schwannomas, as well as some cranial nerve, other cranial nerve lesions. And we're using those as a test, a proof of principle to kind of apply it more widely. And so that is in the works right now. As far as drug targets, for finding drug targets or finding new treatments for hearing fluctuation and Meniere's disease. So we think that the straight answer is that we don't know yet. We have a mouse model for hearing fluctuation, as well as a human clinical protocol that we're working to get up and running, where we're going to be looking for targets that relate to the stria. So now that we have these atlases of the whole cochlea even and the stria, we can look at differences in gene expression systemically and look for targets that are applicable to the inner ear. So we're beginning to look for those in humans with hearing fluctuation. Now, to get you more directly to treatments and to think about things like immunosuppressants versus antivirals versus steroids, those are things that we use now. In my practice, we don't just use steroids, we use antivirals for some forms of autoimmune inner ear disease and some autoimmune variants of Meniere's disease. We use immunosuppressants, a variety of them, usually in combination with one of my rheumatology colleagues, where we treat and try to stabilize hearing fluctuation in those patients. But what we're really trying to do right now is try to find, we're trying to use the data that we have to find more rational targets and to repurpose drugs that are potentially already out there. I mean, the COVID-19 pandemic is a perfect example of how repurposing approaches are being used to find new treatments. And I think we need to use similar approaches to find treatments for sudden hearing loss and for diseases like Meniere's disease and autoimmune inner ear disease. I think I got to your questions, but let me know if I didn't answer them specifically. And I'm sorry I don't have a hugely specific answer. I can tell if you want to talk more about immunosuppressants, that can be
Yeah,
used.
well, my only experience with a few patients is methotrexate, but I don't think it's worthy for their fluctuation to use methotrexate with the side effects that it has. So I don't know if you have other experience with other immunosuppressors. Yeah, which ones do you use?
So have you used, so we've used tofacitinib on some of my patients. It's a JAK kinase inhibitor.
Which is the name? Can you please say it again?
It's tofacitinib. It's TOF.
TOF.
I always misspell this one. So let me just okay, TOFACIT INIB. Okay. The reason we've used that in some of my patients, so a lot of the immunosuppressants require infusions. like, you know, subcutaneous injections. So I have some of my patients who are on things like that, like Rituxan and that kind of stuff. But tofacitinib is an oral medication. It's the first of its generation, so it can be taken at home. You know, and some of the newer biologics are becoming oral. So those are some things to look at. And I also want to be very clear that I don't do any of this without. I have a very active collaboration with several rheumatologists. So I work with them and make suggestions, but I let them kind of guide that treatment. But I do keep very up to date on kind of what's available and what the evidence is behind it. And sometimes you don't have good evidence. So we still treat some of our patients with methotrexate. And sometimes it's marching through different immunosuppressants to try to find one that works. Something simple from the beginning, though, is I do use famciclovir for some of my patients. And I use it, it's not just a 10-day course. I have them on it for about at least 3 months. And we load them up and then we kind of have them on a maintenance dose. So that is something that I do myself. And it's something that Jennifer Derryberry has described for kind of autoimmune inner ear disease patients, as well as some Meniere's patients with autoimmune variants. So
okay, thank you.
Hopefully that helps.
All righty. Well, anybody else have any questions? Mike's got about 5 more minutes. He's got a meeting in about 5 minutes. So if there's anybody else that has a question, please ask it. I think the lecture today brought us to a different level of in our conferences here, because everything's been very clinically oriented. This is definitely a subject that most of us don't even look at in our practices, and I think that it's probably opened some of our eyes to, you know, where the future of a lot of things are with respect to otology, especially sudden sensorineural hearing loss, and, you know, presbycusis with respect to what kind of treatment will there be in the future. Mike, do you have any comments on that, on either one of those or both?
So we think that I think we're taking kind of we're looking at how things change in response to different things like cisplatin, and we've got kind of some hearing fluctuation studies that we're undertaking right now. But I think a lot of the targets that we identify in the Stria are going to relate to a lot of different mechanisms, and any drugs that we identify may be possible treatments for other kinds of diseases of the Stria. So for instance, diseases where we think, or drugs that we think might be applicable in the case of cisplatin might also be applicable in sudden hearing loss, and might also be applicable in presbycusis. So I think there are just a lot of things that we don't know, and I think we need to take the data that we find and the application of the data that we find from looking here to doing actual trials in humans. And that's part of what my group is going to be doing in the next couple of years. We're transitioning to kind of building human trials.
Great. Any other questions? All right, Mike, listen, thank you very much for your time. Anybody who has any questions that they might think of later, please put them on the WhatsApp platform and we'll get them to Mike and we'll get you the answers, okay? In the meantime, look forward to seeing you guys on Thursday. We have Dr. Bill Lawson who happened to have been my professor at Mount Sinai Hospital in New York City. He's going to talk about auricular reconstruction. I highly recommend this lecture, okay, because he's truly a master and a Michelangelo. So listen, thank you everybody for joining. Mike, thank you again for taking your time out, and everybody have a pleasant day and tomorrow.
Thanks everyone. See you.